For this particular lesson, we're starting in the second half, Unit 2 of the Diversity of Living Things in our Science Fusion textbooks. This Unit 2, Lesson 1, covers archaea, bacteria, and viruses. Our essential question, if you're following along, is on page 78. What are microorganisms? By the end of this lesson, you should be able to describe the characteristics of archaea, bacteria, and viruses, and explain how they reproduce or replicate. So we want to know, what is a prokaryote on page 80. All living things are either prokaryotes or eukaryotes. If you look here on the left, those are prokaryotes. They're unicellular or one-celled organisms. There's no nucleus. The DNA is just floating in the middle of the cell. And over here we have eukaryotes with a defined nucleus, membrane-bound organelles. This is eukaryote, and this is our prokaryote. So we're looking today at our prokaryotes. Eukaryotes are made up of one or many cells, each of which has a nucleus enclosed by a membrane. Prokaryotes do not have a nucleus or membrane-bound organelles. Almost all prokaryotes are single-celled. Prokaryotes can be seen with a microscope only, so they're called microorganisms. Prokaryotes are divided into two domains, bacteria and archaea. Bacteria is a domain of prokaryotes that usually have a cell wall and usually reproduce by cell division. Archaea is a domain of prokaryotes that are genetically very different from bacteria and that have unique chemicals in their cell walls. Although they are very small, prokaryotes can get energy and reproduce, and many can move. So what are some characteristics of the archaea, following along on your textbook on page 81? Archaea are organisms that have many unique molecular traits. Archaea are prokaryotes, but their cell walls are chemically different from those of bacteria. Some molecules in archaea are similar to molecules in eukaryotes. Some molecules in archaea are not found in other living things. Archaea often live where nothing else can, such as very hot springs and in extremely acidic or salty habitats. Archaea flourish near deep-sea vents where no light reaches, and they can use sulfur to convert energy. So what are some characteristics of bacteria on page 82 in your text? Most known prokaryotes are bacteria. The domain bacteria contains more individuals than all other domains combined do. Many bacteria are consumers that get nutrients by feeding on other organisms. Some bacterial consumers are decomposers, which feed on dead organisms. Other bacterial consumers live in or on other organisms. Bacteria that make their own food are called producers. These bacteria use energy from the sunlight to make food or glucose and are often green. They use a process called photosynthesis to complete this. Bacteria are small, single-celled organisms, or unicellular. Some bacteria stick together to form strands or films, but usually each bacterium still functions as an independent organism. Each bacterium must take in nutrients, release energy from food, get rid of wastes, and grow on its own. Most bacteria have a rigid cell wall that gives them their shape. Each shape helps bacteria in a different way. Bacteria that are shaped like spirals can move like corkscrews. Bacteria shaped like rods quickly absorb nutrients. Round bacteria do not dry out quickly. Bacteria can be found almost anywhere on Earth. They can be found breaking down dead material in soil, making nitrogen available inside plant roots, and breaking down nutrients in animal intestines. Some bacteria can survive during periods when the environmental conditions become harsh by forming endospores. An endospore is made up of a thick protective coating, the bacteria's genetic material, and cytoplasm. They can survive in hot, cold, and very dry places, and when conditions improve, the endospores break open and the bacteria become active again. So how do bacteria reproduce? I'm on page 84 and 85 in your text. Both archaea and bacteria reproduce by what is called binary fission. Binary meaning two, or splitting in two. Fission means splitting. So binary fission means to split in two. This is where reproduction in which one single-celled organism splits into two. Here we have the original strand of DNA inside, which duplicates. The strands separate. You get your indentations here, and eventually the organism splits into two, like we have here. The first step in bacterial reproduction is copying the cell's genetic information, which is in the form of a long, circular strand of DNA. This loop, called a chromosome, is copied. Then the two chromosomes separate, with one on each side of the cell. The cell's membrane then starts to grow inward, separating the two halves of the cell. A new cell wall forms and separates the two new cells. At the end of binary fission, there are two identical bacterial cells, each with identical DNA. This type of reproduction, in which one parent produces genetically identical offspring, is called asexual reproduction. So how do they exchange this DNA? There are three ways that bacteria can acquire new genetic information. The first is through transformation, then transduction, and then conjugation. Transformation occurs when bacteria take up DNA from the environment. Transduction happens when a virus injects DNA into the bacterium. Some bacteria have a second loop of DNA, smaller than the main chromosome, called a plasmid. During conjugation, two bacteria temporarily join together and a plasmid transfers from one bacterium to the other. The bacterium that gets the plasmid now has new genes to use. They have images of this particular conjugation on page 85 in your text. What are some characteristics of viruses? Okay, so now we're moving away from bacteria and we're going to focus on viruses. I'm on page 86. A virus is a microscopic particle that cannot replicate on its own. That is important. It cannot replicate on its own. It either gets inside a cell or injects a cell with its DNA, often destroying the cell. So in order to replicate, it either has to get inside the cell or inject that particular cell with its DNA, which in turn destroys the host cell. Many viruses cause diseases such as the common cold, flu, and acquired immune deficiency syndrome, otherwise known as AIDS. Viruses can infect people, plants, animals, and prokaryotes. Are viruses living? Viruses contain genetic material and protein, but do not perform any life functions. Viruses do not use energy from nutrients, do not maintain homeostasis, cannot grow, and do not respond to stimuli such as light, sound, or touch. A virus cannot function on its own. It needs that host cell. It can replicate only inside the cell it infects. Thus, viruses are not living. Repeat. Viruses are not living organisms. Okay, we're going to talk about the flu for a minute here, which is on page 87 in your text. Most influenza viruses are not very harmful. When a new virus spreads to many people around the world, however, it can cause what's called a flu pandemic. Pandemic means it pans the world. In 1918, the flu was caused by one of the world's deadliest viruses, which killed more than 50 million people and infected more than 500 million. New strands of the flu virus are constantly developing. Scientists monitor these outbreaks closely so that vaccines can be made. So how do these viruses replicate? We're on page 88 in your text. Viruses can replicate only inside a living cell that serves as a host, which is a living thing that a virus or parasite uses for resources or shelter. Viruses attach to specific types of host cells and invade them. Many viruses cannot be spread from one type of organism to another. When a virus enters a cell, or when its genetic material is injected into a cell, the virus takes over the host cell, which follows instructions in the viral DNA. The host cell then accidentally replicates the viral DNA and makes new protein parts for the virus. Then parts of the new virus assemble in the host cell. When the host cell is full of new viruses, they burst from the host cell. This step is called lysis, which kills the host cell. The new virus searches for new host cells, and the lytic cycle, which causes lysis, begins again. Some viruses insert their genes into the host cell, but the new viruses are not made right away. The genes can stay inactive for a long time. When the genes do become active, they begin the lytic cycle and make copies of the virus. Here we have the virus. It attaches to the host cell, injecting its DNA. The host cell replicates that DNA, causing multiple viruses to be cloned. Then, eventually, when there's more viruses than the host cell can handle, the host cell lyses or breaks, and all the viruses